Welcome. In the last session, I discussed about Mendel's monohybrid cross, and now in this session, I'm going to discuss about test cross, and then later we will see Mendel's dihybrid cross. Okay, so first, let's see what is test cross. Right, so here we know in monohybrid cross, after crossing the parents, he obtained all tall peak lines. Right, so to know whether the F1 generation is homozygous or heterozygous, we need to cross that F1 generation with the recessive parent. Then we can come to know whether it is heterozygous or homozygous. So I'll just repeat that monohybrid cross here. So here homozygous tall pea plant is crossed with homozygous recessive pea plant. So we know, okay, so we obtained the gametes, and now these two, capital T small t, that is F1 generation, we have seen that all 100% are tall pea plants were obtained. To know this F1 generation, whether it is a homozygous or heterozygous, we need to cross this one with its recessive parent. Means, so we need to take this one and then cross with its recessive parent. So this is the recessive parent, and this one will be the dominant parent. So this is dominant parent and this is recessive parent. Okay, so we'll see that one. So before that, let me tell you the definition of test cross. So is a cross between the individuals of, is a cross between individuals of unknown genotypes. Okay, cross between individuals of unknown genotype and its recessive parent is what we call test cross. Of course, Mendel, what he did is he has taken a tall pea plant from the F2 generation. So he has taken a tall pea plant from F2 generation and then he crossed with its recessive parent. Okay, a tall pea plant is crossed with the, of course, a tall pea plant, okay, a tall pea plant from F2 generation. So from F2 generation was crossed with the recessive parent, crossed with the recessive parent. So that is what Mendel did. Okay, so Mendel crossed a tall pea plant from where F2 generation. So he has taken from F2 generation and then crossed with a recessive parent. So this is what I have shown here, F1 generation. Of course, in case of Mendel, he has taken from F2 generation. So now let's see how the test cross will be. So capital T small t, so that will be from F2 generation and crossed with the small t small t. So this is homozygous dwarf plant. So this is a recessive parent, is recessive parent, and this is from F2 generation. So this is a tall pea plant. So these will be the parents. Okay, now during the formation of gametes, so here this tall pea plant, which is heterozygous, actually forms two types of gametes in equal proportions. So this is, okay, so 50% of gametes will have capital T and 50% of gametes will have small t. Whereas this recessive parent will form, okay, only one type of gamete that will have, okay, the recessive character. So these are gametes which are haploid. Okay, so now if this gamete comes in contact with this, then capital T small t, then it is tall pig plant. And if this one and this one comes in, okay, then this is dwarf pea plant. So Mendel obtained okay, the tall pea plants and the dwarf pea plants in 1:1 ratio. So 50-50 okay, percent he obtained the tall pea plants and dwarf pea plants. So let's we put it in the Punnett square here. Okay, so male gametes and female gametes. So capital T, small t, and here we have only one type of gamete from one of the parent. So here capital T, small t, and small t, small t. So this will be the tall, and this will be dwarf. So now taking the phenotypic ratio. So taking the phenotypic ratio of monohybrid test cross. So phenotypic ratio of monohybrid, okay, test cross. Then here in this case tall is 1 and dwarf is 1. So here the ratio will be 1:1. Then coming to the genotypic ratio of monohybrid test cross. So genotypic ratio of monohybrid test cross. So when we, okay, take this, okay, so here how many heterozygous we have? Okay, heterozygous tall is 1. So we have heterozygous tall 1 and okay, homozygous dwarf is 1. So heterozygous tall 1 and homozygous dwarf 1. So hence the ratio will become again 1:1. So here for the test cross of monohybrid, right here both genotypic and phenotypic ratios will be 1:1. Okay, so this is how we can find out the genotype of an unknown individual. So this is what we call test cross. So Mendel performed the test cross to know whether the individuals in the F2 generation whether they are homozygous or heterozygous, and he came to know that they are heterozygous. And the dwarf character, it did not express in the F1 generation. It was hiding in the F1 generation, but got expressed in F2 generation after, okay, self-crossing the F1 generation. Right, so this is about the test cross. So now let's move on to the next one, the Mendel's dihybrid cross. So we have studied till now inheritance of only one gene. Now we will see inheritance of two genes. So inheritance of two genes. So here the inheritance of two genes, which I also call it dihybrid cross, also called dihybrid cross. And how we define this, okay, is a cross between two plants involving two traits. Okay, a cross between two plants involving two traits, then it's called a dihybrid cross. So all the remaining characters will be same except for two, so those will be different. Now let's see what that characters Mendel have considered, right? So one, it is seed color. So this is one trait what he considered. Then another one, seed shape. So seed color and seed shape are two traits that Mendel considered. So here seed color, in case of a pea plant, we know there are two alleles for this. One is yellow color and another one is green color. So yellow color and green color are two alternative forms of this trait seed color. Then coming to the seed shape. So in this case of seed shape, right, there are again two alternative forms. One is round and another one is wrinkled. Round and wrinkled. For seed shape, round and wrinkled. For seed color, yellow and green color. And of course, after completing the experiment, Mendel came to know that yellow is dominant. So hence I'm going to take it with the capital Y. And its alternative form, the green color, I need to take with the same alphabet, but I need to take small letter. So this is dominant and this is recessive. And in this case of a seed shape, so here we find the round is dominant, so I'm going to take capital R. So this is dominant and this is small r is recessive. Right. So these are the two traits that Mendel considered for his dihybrid cross. So now what he has done is he crossed a homozygous round yellow. So he crossed a homozygous round yellow pea plant with the so crossed homozygous round yellow pea plant with the homozygous wrinkled green pea plant. So what Mendel have taken is pure breeding pea plants, one with homozygous round yellow condition and another one with wrinkled green condition. So that's what I'll mention here. So capital R, capital R. So we need to take we know it's the parents will be diploid and hence we need to take two genes for a character, capital R, and capital Y, capital Y. So this is homozygous round yellow. So you can consider homozygous round yellow crossed with small r, small y, small y. So this will be homozygous wrinkled green is homozygous wrinkled green. So this is what they have taken, the homozygous round yellow. That's what I mentioned. Homozygous round yellow is crossed with the wrinkled green pea plant, right? So here these are parents. So 2n, parents, right, 2n. Now the gamete formation, meiosis, and this results, we know out of these two genes only one gene will be entering the gamete, and out of these two only one. So that is it will become capital R, capital Y. So here it is diploid, but we know gametes are haploid and gametes will receive only half the number of genes, okay, from the parents. Right, now here also same, meiosis, and out of these two, one small r and one small y, and gametes haploid, right? And fertilization between these two gametes will result in the formation of capital R small r, capital Y small y, and this is F1 generation. So what he observed in the F1 generation is that he obtained all round yellow. So he obtained, so 100%. So if he collected some 1500 seeds, all the 1500 seeds, when they raised, developed okay, round yellow features. Okay, now we know the next step of Mendel is self-crossing the F1 generation. So we need to, okay, self-cross the F1 generation. So the next step is self-crossing F1 generation. So self-crossing the F1 generation. So here that will be the capital R, small r, capital Y, small y, again into capital R, capital Y, small y. So these will form the parents to the next generation, right? So this is round yellow, and this is also round yellow. Okay, right? Of course, when we are doing, or Mendel is doing these experiments, he do not know that these the yellow color, of course here the recessive, this is the green color, and okay, the wrinkled condition, so they are hiding, so they did not express in the EF1 generation. So because of that, we call them recessive, okay, characters. The one who are expressed, they are dominant. Okay, so here, so he has taken from the EF1 generation and self-crossed the EF1 generation. So now let's see the formation of gametes. So here uh, most of the students will go wrong. Okay, just I'll make here a difference between, okay, capital R small r and capital Y small y. So this makes us, okay, to draw the gametes, right? So here you can take, right, this capital R and capital Y. So capital R, capital Y will be one gamete. Then capital R small y will be the next gamete. Then the small r capital Y will form another gamete, and small r small y will form another gamete. So from this parent we get totally 4 gametes. So here also the same situation. So capital R, capital Y is one gamete. Then capital R small y is another gamete. Then small r capital Y, okay, small r capital Y is another gamete. And lastly, small r small y is another gamete. So these are gametes, right? Already by drawing these lines here, it has become cumbersome, and now, okay, drawing, okay, further lines. So here this one and this one, capital R, capital R, capital Y, capital Y, so it will be quite difficult. So hence I'm going to put these gametes in Punnett square and see that what will be the F2 generation. So let's see. right? So on one side the male gametes, so let's take capital R, capital Y, okay, then capital R, small r, capital Y, small r, small y. So here also the same gametes, so both male and female gametes are same, okay. So because it's a self cross, so capital R, y, small r, capital Y, and small r, small y. Okay, so let's see the possibilities. So capital R, capital R, capital Y, capital Y. So we get here. Then the next one, capital R, capital Y. So this and this, then capital R, small y. Now this one with this one. So capital R, capital Y, capital Y. Then this one with this capital R, capital Y, small y. Okay. The next one, this one with this one, capital R, capital Y, small y. Capital R, small y. And this one, capital R, okay, small r, then capital Y, small y. Then capital R, small y, small y. Okay. The next, this one comes, so here capital R, small r, then capital Y, capital Y. Then capital R, capital Y, small y. And this one, small r, capital Y. And small r, capital Y, small y. So the last row, so this one with this capital R, capital Y, small y. Then the next one with this capital R, small y, small y. And this and this, small r, capital Y, small y. And lastly, small y, small y. Right. So now let's see. This is round yellow. So this is round yellow. This is also round yellow. This is also round yellow. And this one is also round yellow. Okay. And coming here down the second row, the round yellow again. Okay. Now it's not round yellow. This will be, okay, round green. This one is round yellow again, and this is round green. The next one is again round yellow. The next one is again round yellow. And here we can see both are small r's. It means it is wrinkled. It is wrinkled, and we can see capital Y, so hence it is wrinkled yellow. And in this case also we get wrinkled yellow condition, right? And coming to here, the last row, again we can see here round yellow, and this is round green, and this will be wrinkled yellow, and lastly it is wrinkled green. Okay, so this is how we get, okay, the Punnett square. Now from this we need to, okay, draw the phenotypic and genotypic ratios. So first we'll see the phenotypic ratio of dihybrid cross. So phenotypic ratio of dihybrid cross. So what will be the phenotypic ratio of dihybrid cross? So we'll see here. So how many we have is round yellow, okay, then round green, then wrinkled yellow, and lastly wrinkled green. So we need to find out how many are all these round yellow. So you can see here the round yellow. So here it is 1. Okay, so this is 1. Okay, then this is the second one, third one, 4, okay, 5, then 6, then 7, then 8, and this is 9. Okay, so totally we find round yellow will be 9. Then coming to round green. So coming to round green, so you can see round green, here it is 1. Okay, then this is the second one, and this is the third one. So here round green are 3 in number. Then wrinkled yellow, so this is 1, 2, and 3. So wrinkled yellow is 3, and lastly wrinkled green is only 1. So the ratio, that is a phenotypic ratio, is the phenotypic ratio of dihybrid cross will be 9:3:3:1. So that is the phenotypic ratio. Okay, now let's look at the genotypic ratio of dihybrid cross. So genotypic ratio of dihybrid cross. So in this we need to find out, okay, here, okay, capital R, capital R, capital Y, capital Y. So how many we have, okay, of them? Capital R, capital R, capital Y, capital Y. So we need to find out how many here they are present, right? So here its number, so you can see nowhere in the table same capital R, capital R, capital Y must be only present. So with this genotype you don't find anywhere else. So hence the individual with this genotype is only 1. The next one is a different genotype. So capital R, okay, capital R, capital Y, small y. So here we need to find out is there any other genotype with the same capital R, capital R, capital Y, small y. So no, here also you don't see. Then you can see here, okay, these two are, okay, capital R, capital R, capital Y, small y, same genotype. So nowhere else you will find the same genotype. So in this case, okay, so I'm going to take this capital R, capital Y, small y as 2. The next one will take capital R, small r, capital Y, capital Y. So coming to this, how many do we find here? So this is 1, okay, and you can see here this is another 1. So 1 and 2 with the same genotype, and nowhere else you will find with the same genotype in this Punnett square. So hence I'm going to take this as 2. Next coming here, capital R, capital R, capital Y, capital Y. So how many we have? Okay, so this is 1, okay, 2, this is 3 and 4. So you can see all this on this line you will find, okay, capital R, small r, capital Y, small y. So that it will be 4. So its number is for capital R, capital R, capital Y, capital Y. Then the next one, capital R, capital Y, small y. So we already finished it. Then we'll see capital R, small y, small y. This we did not finish. Capital R, small y, small y. So how many do we find in this Punnett square? Okay, right. So nowhere else I think there is another with the same genotype. So it will be only 1. Will be only 1. Then next one we finished already. Then the next one, capital R, small r. So this we did not finish. Capital R, small y, small y. So do we find anywhere else? So this one and this one, you can see capital R, small y, small y. Here also capital R, small y. So this will be okay 2 in number. Then capital R, capital Y. So this genotype we already finished here. Okay. Then this also capital R, capital Y, small y we finished. Then the small r, capital Y. So this genotype, do we have anywhere else? No, it's the only one. So hence I'm going to take it 1. The next one, that is this also we did not finish. Okay, so I'm going to take, I think, a little bit. Right. Okay, so I have written on that side. Next one, so we have finished this one. Then the next one, small r, capital Y, small y. So this small r, capital Y, small y, you will see it again here. So it is okay 2 in number. Then this genotype we already finished it. Then capital R, okay, small y also we finished it. Then small r, capital Y, small y also we finished. And lastly we have is small r, small y will be 1. So the ratio, that is genotypic ratio, will be 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1 is the genotypic ratio. So and if you add, you should get 16. Okay, 3, 6, okay, 7, 11, okay, 13, 14, 15 and 16. So total you have okay 16 genotypes. So here in this table the total possibilities are 16, and here also you will get the same total number of right 16. So that will be, so 9 is to 3 is to 3 is to 1. Okay, so here 10 plus 6, that is 16. You will get total. So when you total it, so if you feel that if you have gone wrong, so just adding them will make you to get okay. If more if you get, then you did a mistake in deriving this phenotypic ratio or in genotypic ratio, and you need to mention as they are. So 9 is to 3 is to 3 is to 1 is the dihybrid cross, okay, phenotypic ratio, and 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1 is the genotypic ratio of dihybrid cross. Okay, so this is all about the inheritance of two genes or dihybrid cross. Right, so my question is find out the phenotypic and genotypic ratios in case of dihybrid test cross. Already I have shown you that how to perform the test cross, and we have seen monohybrid test cross. So if you are able, if you are capable of finding out the dihybrid cross ratio, I mean, so if you have understood it, just try to find out the dihybrid test cross ratio, right? Now after these experiments, Mendel have given some laws, so we will see about those, okay, Mendel laws. So we'll see about Mendel laws. Okay, so again I'm repeating, try to find out the dihybrid test cross ratio, that is both phenotypic and genotypic ratios. So okay, now let's see Mendel, okay, laws. So here Mendel proposed okay, three laws. We will see. The first law is law of dominance. So it's called law of dominance. Whereas the second law is called law of segregation. The second law is called law of segregation. Also, I can call it law of purity of gametes. So the first law is law of dominance, and the second law, the law of segregation or law of purity of gametes. And the third one we'll see about is law of independent assortment. We'll see it independent assortment. So now we will try to understand these three laws, what actually is law of dominance and law of segregation or law of purity of gametes and law of independent assortment. Okay, so let's see the first one, law of dominance. So what this law states is that uh, only one factor in a contrasting pair, only one factor in a contrasting pair, so only one factor in a contrasting pair, only one factor in a contrasting pair expresses itself in F1 generation. Only one factor in a contrasting pair expresses itself in one generation. So here in Mendel's time there was no word called gene, and no one knows properly what actually is this DNA, and they don't know that how these what will be the physical okay form of these genes, where they are present, how they are present, what they are made up of, nothing. No, even Mendel do not know about it. Okay, so he, of course, he simply said the characters, how they are inheriting from parents to offspring. So that's the thing what he mentioned. And there is no word called gene in the dictionary of biology in those days. So that is in 1865. And moreover, we came to know the complete structure of DNA, okay, the double helix model, everything that was in 1956 during okay, very in 1900s we came to know about the DNA. So in those days, no one knows what actually the gene is. So even Mendel, right? So he simply said in his words something that is okay as a factor. So that gene is actually in his words is a factor. So he used the word factor, okay, because I told you there is no word gene in the dictionary of biology in those days. So only one factor in a contrasting pair. So you can see the contrasting pair, capital T, small t. So this is a contrasting pair, is a contrasting pair. So in this contrasting pair, okay, one of it will get expressed. The one which is expressed is the dominant. Okay, so in a contrasting pair, okay, so this is one factor and this is another factor. Okay, so there are two factors in a contrasting pair, and out of these two factors, okay, only one of it is expressed. So only one of it is expressed. The one that is expressed is dominant, and the other, okay, so the other factor that is not expressed, that is not expressed is the recessive. Okay, so according to the law of dominance, what Mendel states is that only one factor in a contrasting pair expresses itself in the F1 generation. Okay, and he mentioned a few points on this. So that characters are controlled by discrete units. So characters are controlled by, okay, discrete units called factors, are controlled by, okay, discrete units, uh, okay, called factors, right? So here that is one point. Then, and he also told that these factors will occur in pairs. So factors occur in pairs. So you can see here the factors are present in pair. Even if you consider, okay, capital R, okay, capital Y, right? So this is a contrasting pair, okay, in which, okay, only one of the factor is expressed. Okay, so we know, of course, this I'll take it later. So factors occur in pair. So factors occur in pair, and another one, okay, in a dissimilar pair. In a dissimilar pair of factors, in a dissimilar pair of factors, okay, one member of the pair, one member of the pair dominates. So one, okay, member of the pair dominates. So in a dissimilar pair of factors, one more. So here these two are dissimilar, and out of these two, one member will be dominating and other one will be recessive, right? So that's, you can take capital R, small r in the case, or capital Y, small y, right? So that's what the factors will occur in pairs. Okay, so they occur in pairs, and in a dissimilar, so these two are different. So you can see in the tall and dwarf, okay, round and wrinkled, yellow or green, only one member will dominate. So that's about the law of dominance, right? The next one, we'll see about law of segregation, and also law of independent assortment in the next session. And after Mendel, many others followed Mendel. So how he did experiments, they also have done experiments, but they did not get the result like Mendel. So what is that, we will see in the next session.